thank you for joining us today, Dr. Zhang. It's a pleasure to speak with you. To start off, can you tell us a little bit about your current research goals?
My current research goals are mostly focused on alternative splicing and how it's regulated in the biological system and to achieve the better fitness for different organisms. So splicing is something that has been discovered for decades, and then I think it has been put at the back burner for many, many years. And then now, because of next generation sequencing, it becomes harder. So people started to look at it, and then I have been working with splicing since 2006. So I do appreciate a lot, and so I'm happy to see the change, and then thinking how my research can contribute to that. Specifically, now I'm looking at a protein called SR45, which is now named as a splicing activator. So which means if you have this protein, proper splicing could happen more frequently. And otherwise, you may have more alternative splicing that could lead to different products in protein. So now I'm interested in seeing what protein network that SR45 is working with. Because right now, it's not very clear. There are different candidates, different groups have come up with. We also did. And then there's still very much of not very clear idea or proposal as to how SR45 is involved. So I want to learn more about that. And on the other hand, it's more of downstream kind of outcome that SR45 can work with, is what type of gene, I guess, pathways or genes SR45 directly or indirectly regulate. So I'm looking for a network, either at partner level or the downstream targets.
Interesting. And you mentioned there's been this renewed interest in RNA splicing with the availability of next-gen sequencing. Mm-hmm. Can you talk a little bit about how you are using next-gen sequencing in your work?
Yes, absolutely. Actually, in 2004, that was the beginning of omics for me. So at the time, microarray was still pretty much the only way to study transcriptome. But lately, since RNA-seq becomes more and more, I guess, affordable and popular, there are more analysis tools available. So we're now using RNA-seq to look at what RNA targets that can be changed when you have or you don't have SR45. And then that's one thing. The other is for proteomics level, we're also interested, but we haven't done a lot just because the specificity and the optimization takes time. But overall, we want to see at omics level to see how SR45 can cause a change. And definitely RNA-seq right now is something we are doing.
And so how have you used DNASTAR software in that work?
We have used it quite a bit for several years, actually. So especially the cloning suite, we have used very frequently, especially for students to do some design or understanding what happens. And then we also use the protein 3D, and then that helps us to visualize how protein site groups and then how they orientate, and then if they change, what it looks like for protein. And then just starting from the end of last year, we started to test on the Sigma engine and the ArrayStar, and that worked quite well, I have to say. I really like it. It takes time just because any kind of tool like this is going to take time. There is a learning curve, but I think all the videos and the webinars really helped on your website. And so just by watching those, I was able to use Sigma engine and ArrayStar try to analyze my results.
That's interesting. It sounds like you're really using all the different branches of our software.
I think so.
Do you see yourself continuing down that path?
For the next gen stuff, it's only on the transcriptome level. So I want to see how much of this change can be translatable into protein. So if we were going to identify some targets or interesting candidates we want to follow up at protein level, we want to see how splicing affects the protein folding and structure and then therefore function.
Is there anything else you'd like to share?
I think there is one particular thing I just would like to share is I work at primarily undergraduate institutions. So I only work with undergrad in another word because we just don't have graduate students. And for a lot of other I guess PIs, when they're in those RO1 institutions, they would say, "Oh, of course, I work with undergrads too." But it's different. You have postdocs, you have grad students, you have technicians. We don't have any of them. We have to be all of them. So that's very different. That's why I'm really relying on inventing the approaches, protocols, and looking for tools that are undergraduate student friendly. And I think your cloning suite definitely is one of those examples. And the Protein 3D viewer, it's fantastic. Because for students, undergrads, to visualize proteins is very hard. But Protein 3D it's just fantastic. It's easy to use. And then they can see it right away, and then they can rotate, change things on their own. So I don't have to teach them much. And then they have fun. That's most important. I think because of that, some of my students, they just get so interested in proteins, and then they go to grad school or med school. They decided to follow up and then keep working on proteins.
To see more interviews with DNASTAR users, please visit our website, DNASTAR.com. And to have your work featured on DNASTAR LabViews, contact us at DNASTAR-info@DNASTAR.com.